So Chapter 11 is going to talk about cell signaling by chemical messengers. So here we have the learning objectives. Make sure you can answer all of these completely by the end of the lecture. So we have several different types of chemical messengers. We got hormones, neurotransmitters, cytokines, retinoids, growth factors. Some chemical messengers can bind intracellularly, some can bind on the surface. So just be able to tell me all the different types of examples. If I ask you, what is an example of an intracellular receptor? Then nuclear hormone would be an example. If I ask you what are some type of surface binding receptors, then we have ion channels, tyrosine kinase receptors, and G protein-coupled receptors. So expect some of these questions. So you should be able to tell me how chemical messengers work. There's 3 key steps. So the first is the secretion of a chemical message. So just take a look at the picture. You have a secretion of a stimulus to the cell, and then step 2 is the binding of a message to the cell surface receptor. So you see those chemical messengers binding to either a plasma membrane receptor or going through diffusion. So in step 3, you can either have diffusion of the hydrophobic message across that plasma membrane, or again, it can go onto and bind a plasma membrane receptor. So once it's inside the cell, it'll go through signal transduction, which will provide a response. Alright, so this slide shows you some typical chemical messengers in the body. So we have the nervous system, and really there's 2 types of messengers. So we have the small molecule neurotransmitters called biogenic amines, and then we also have something called neuropeptides. So those are examples of nervous system chemical messengers. We have endocrine system chemical messengers. So some examples of those are polypeptide hormones such as insulin, steroid hormones, thyroid hormone, and retinoids. So no need to focus on the structure, but know the examples of the types of chemical messenger systems. So continuing from the previous slide, we have several other chemical messengers. Some are from the immune system, so those are your cytokines and your chemokines. Then we have our eicosanoids, remember your short-term signaling molecules. And then we have growth factors. So chemical messengers are often classified as either endocrine, paracrine, or autocrine. So it's important to know the difference between the 3. So endocrine are distant targets. So if you look at picture A, you see that a hormone is secreted into the blood and it travels to a target cell. So that's going to be a distant target. So an example is estrogen hormone. Paracrine are local targets. So think of your neurotransmitters such as acetylcholine. So in picture B, you see 2 different cells, but they're adjacent to each other. So local targets are going to be paracrine. And then you have autocrine, which is self. So it's just involving a messenger that acts on the cell from which it is secreted. Okay, so this is another learning objective, knowing the difference between intracellular and plasma membrane receptors. So when we talk about receptors, they usually fall into 2 categories, intracellular or plasma membrane receptors. So messengers that are used intracellular have to be hydrophobic molecules. And if we're talking about the plasma membrane, then they're going to be receptors bound by polar molecules. So plasma membrane, polar molecules, such as peptide hormones, cytokines, catecholamines, and intracellular receptors are going to be hydrophobic molecules. So steroid hormones, thyroid hormone, retinoic acid. Know the differences between them, and then also know the examples. So in this slide, we're talking about those intracellular receptors. Remember, intracellular is hydrophobic molecules. So this specifically focuses on the steroid hormone, thyroid hormone superfamily. So most of these steroid hormones are found in the nucleus. So they're nuclear hormone receptors. RAR, TR, VDR, and AR are all of the different types of receptors found there. So you should know what RAR stands for. TR, VDR, and AR. So that's all shown below. If you look at the picture on the right, There's also several examples of some of those hormone receptors, so you should know those examples, but you shouldn't have to worry about the structure. And then sometimes these hormone receptors can bind to RXR, which is 9-cis-retinoic acid, and it forms a heterodimer. So this binds to DNA and activates transcription. So this is learning objective 4, knowing the major classes of the plasma membrane receptors. So here we have 3 different classes of plasma membrane receptors. The first one is ion channel receptors. So these are small molecule neurotransmitters. The example that he's going to provide is acetylcholine. So we also have receptors that are kinases, or that bind kinases, and then we have heptahelical receptors. So we'll talk about each of them, starting with the ion channel receptors. So an example of an ion channel receptor is acetylcholine receptors on the neuromuscular junction. So these work at the nicotinic acetylcholine receptors. So first we have to understand everything in the picture. The yellow portion is your presynaptic nerve terminal. If you notice inside of it, we have acetylcholine synaptic vesicles. So these vesicles contain the neurotransmitter acetylcholine. So our job is trying to release them from those vesicles so that they can bind to their receptors. At the very bottom, we have a muscle cell. So these muscle cells have the acetylcholine receptors, and then they also have voltage-gated sodium channels. In the space underneath the presynaptic nerve terminal is the synaptic cleft. So that's where acetylcholine will have to travel to get to its acetylcholine receptor. And so how this all begins is first we need an electrical stimulus. So your central nervous system will send some sort of an electrical signal that's called an action potential. So as soon as that action potential arrives at that presynaptic nerve terminal, it's going to trigger a few things. So the first thing that's going to happen is your calcium channels on that presynaptic membrane is going to open. So that's going to lead to an influx of calcium. And this influx of calcium is going to cause all those acetylcholine synaptic vesicles to fuse into the presynaptic membrane. So as soon as those acetylcholine synaptic vesicles fuse to the presynaptic membrane, acetylcholine is released into the synaptic cleft, so that blue area. So once acetylcholine is released, it can bind to its acetylcholine receptor. And once that occurs, you're going to have your muscle contraction. So that's going to trigger another action potential, but on your muscle cell. So here we're still talking about the acetylcholine neurotransmitter. This is just zooming in a little bit. So remember, it acts on the nicotinic acetylcholine receptors. So the receptor is composed of 5 subunits. If you look, there's only 3 to keep things simple. There's 2 alpha and 1 gamma subunit. And then the green parts, there are 2 acetylcholine receptors bound to it. So as soon as 2 acetylcholine molecules can bind to their acetylcholine receptor, it's going to cause a conformational change which opens up this gated channel. So once the channel is open, the sodium ions are going to diffuse in and the potassium is going to diffuse out. So the second class of plasma membrane receptors are receptors that are kinases or that bind kinases. So there's going to be 3 that we're going to talk about. The first one is tyrosine kinase receptor, then we have JAK-STAT receptor, and third is serine-threonine kinase receptor. So we'll go through all of these in the next few slides. So the first one we're going to talk about are tyrosine kinase receptors. So first of all, those yellow things are monomers with a single membrane spanning helix. So if you look, you can see a membrane spanning helix in each one of them. So they don't become a homodimer until a certain ligand binds to it. So in the example, growth factor is the ligand that binds to it, which makes both of the monomers converge together and becomes a homodimer. So now that it's a homodimer, those pink boxes, which are called tyrosine kinase domains, and remember, kinases are things that phosphorylate something else. So those tyrosine kinase domains are going to phosphorylate each other. So that's represented by the P. And once your tyrosine kinase receptor is phosphorylated, another protein will come on by and take its phosphate. So SH2 domain is a certain type of transducer protein that's going to take the phosphate and bring it somewhere in the cell, which will trigger a cellular response. So again, you have monomers in the membrane. A ligand will bind to it. In this case, it's growth factor. Once that happens, those two monomers are going to become a homodimer, and your tyrosine kinase domains are going to phosphorylate each other. So now you have an activated tyrosine kinase receptor. A protein from somewhere in the cell is going to come, take the phosphate, and bring it somewhere else in the cell to cause a cellular response. So the next one is called JAK-STAT receptor. It's very similar. Instead of a growth hormone, this time it's a cytokine. So those are cytokine receptors. So as soon as cytokine binds to it, it'll cause both of those monomers to form a heterodimer. So remember, JAK-STAT is a heterodimer, tyrosine kinase is a homodimer. So on these cytokine receptors, there's something called JAK, and JAK stands for Janus kinase. So that's just another kinase. Remember, they phosphorylate things. So as soon as cytokine binds and you form that heterodimer, your JAK is going to be activated and it's going to phosphorylate those cytokine receptors. As soon as the cytokine receptors are phosphorylated, something called STAT, which is signal transducer and activator of transcription, will bind to that phosphate. He doesn't really go too much in detail after this, but JAK will also phosphorylate STAT, and then STAT will go off into the nucleus where it's going to initiate transcription. So the last one is the serine-threonine kinase receptor, very similar to the other two. The only difference is that the ligand is a dimer. So two cytokines will have to bind to those monomers, and they're going to form a cytokine dimer. So those monomers, again, they're forming another heterodimer, and then you'll notice those pink boxes are your serine kinase domains. So again, kinases phosphorylate things, so it's going to phosphorylate the cytokine receptors, and that'll cause another signal transducer protein called SMAD to bind to the phosphate and bring it to somewhere in the cell, causing a cellular response. Alright, so the third and final class of plasma membrane receptors are heptahelical receptors. So they also go by the name G protein-coupled receptors, and they're the most common type of receptors in the plasma membrane. So they are a 7-membrane spanning alpha helix, and they usually work through secondary messengers. So we'll talk about this in the next slide. So here's just a general outline of how these heptahelical receptors work. So you should know those examples. He really does focus on adenocyclase and cyclic AMP. But if you look at the picture on the right, you have a first messenger. So it could be a hormone, it could be a cytokine, it could be any type of neurotransmitter. So in this case, it's a hormone is your first messenger. That messenger is going to bind to your heptahelical receptor on the plasma membrane. And then once it binds to that, it's going to activate a heterotrimeric G protein. So that alpha, beta, gamma with the GDP is going to be activated, and it's going to activate another enzyme, which will further activate a secondary messenger such as cyclic AMP or DHE or IP3, and then that's just going to cause the cellular response. So again, you have the first messenger binding to the heptahelical receptor. Once that occurs, the heptahelical receptor will activate your heterotrimeric G protein. That will activate another enzyme, and then that enzyme will activate your secondary messenger system, and then from there, that's going to cause the cellular response. So now we'll talk about signal transduction through several different pathways. So here's just what does a signal transduction cascade do. Whatever's in those three boxes are very important to know. So if you have a receptor, it's going to be inactive until a signal activates it, right? So once a signal binds to the receptor, it'll become a signal receptor complex. So now you're active. So just think of like the tyrosine kinase receptors or the JAK-STAT receptors. All those type of receptors are active, and they signal for something to happen in the cell. So signal transduction cascades can lead to coordinating regulation of metabolic pathways by altering activity of specific enzymes. They can change in intracellular ion concentrations, and they can activate gene transcription, leading to expression of specific proteins. So definitely know those three. It will probably test you on what signal transduction cascades can lead to. So we're going to talk about signal transduction through tyrosine kinase receptors. So we talked about them a little bit, but we're going to incorporate them into the RAS and MAP kinase pathway. So we're going to talk about that. We're going to talk about phosphatidylinositide phosphates in signal transduction, and then we're going to talk about insulin receptor signaling pathways. So this is an example of the signal transduction through tyrosine kinase receptors, RAS, and the MAP kinase pathway. So in step 1, we have two monomers. Remember those yellow things? They are going to dimerize as soon as a ligand binds to it. So step 1, growth factor binds to it, and they dimerize. Step 2, autophosphorylation occurs. So remember those pink boxes are tyrosine kinase domains. They're going to phosphorylate itself, so that's autophosphorylation. And as soon as that happens, it's going to open up something called an SH2 domain. So in order to bind to this SH2 domain, whatever protein that wants to bind to it also has to have an SH2 domain. So all of this is in the notes below, so be sure to read. So GRB2 is an example of a protein that has an SH2 domain, so it can bind to that phosphate. As soon as that binding occurs, a conformational change will occur in GRB2, which will open up something called an SH3 domain. So now you need something that has an SH3 domain to bind to GRB2. So SOS is a protein with an SH3 domain, so it's going to bind to GRB2, and that's just going to cause another conformational change. So what's important about SOS is that it is a GEF, so that stands for guanine nucleotide exchange factor. So if you notice, RAS has GDP bound on it. RAS is just a G protein. So SOS can exchange GTP for GDP on RAS, which also causes a conformational change and promotes binding of a protein called RAF. So as soon as RAS and RAF bind together, it's going to initiate the MAP kinase pathway. And this ultimately leads to a cellular response that causes either upregulation of transcription or downregulation of transcription. So the next one is generation of phosphatidylinositide signal molecules. So key things, just take some time to look through the picture on the right. We start off with phosphatidylinositide phosphates. That's known as PI at the very top. And it can serve two different functions in signal transduction. So the first one is that it can form PI4,5 bisphosphate. And that can be cleaved to generate either DAG, which is diacylglycerol, and IP3, which is inositol triphosphate. So again, phosphatidylinositol, or PI, can generate PI4,5 bisphosphate. And PI4,5 bisphosphate can be cleaved to generate two second messengers, diacylglycerol and inositol triphosphate. So DAG and IP3. The second function is that phosphatidylinositol can form PI345 triphosphate. So that just serves as a plasma membrane docking site for signal transduction proteins. So another type is the insulin signaling pathway. So this is just a very general one. We'll go on to the next slide so we can explain it first. So insulin receptor has divergent signaling paths, which means that it can lead to all different types of signal pathways. So we have a dimer, again, made of 2 alpha and 2 beta subunits. Insulin is your ligand that binds to it, which activates everything. So binding of the hormone causes autophosphorylation. So insulin binds to it, and the beta subunits, if you look at the arrows, they're going to phosphorylate each other. So once that occurs, it's going to activate binding sites for IRS, which stands for insulin receptor substrate. So this activated receptor kinase phosphorylates IRS multiple times, which allow for certain binding sites to bind other proteins. So if you look on the left, GRB2, remember, has those SH2 domains, and then PLC, if you go back to the phosphatidylinositide phosphate, you'll notice that PLC is required for making DAG and IP3. And then it also has a binding site for PI3 kinase. So if you go back to look at the phosphatidylinositide signal transduction pathway, you'll notice that you need PI3 kinase to go from PI4-5 bisphosphate to PI3-4-5 trisphosphate. Alright, so to understand this insulin receptor protein kinase B signaling pathway, you first have to understand that phosphatidylinositide signal transduction pathway. So go back and know everything about that before you look at this one. So the first thing that happens is insulin has to bind to the receptors. So remember, that causes the beta subunits to autophosphorylate each other, and that allows IRS to bind to it. So IRS, along with PI3 kinase, so that's from the previous pathway, they're going to bind and phosphorylate PI4-5 bisphosphate, which is not shown here, and it's going to make that become PI3-4-5 trisphosphate. So protein kinase B, also known as AKT, and PDK1 are recruited to the membrane by their pH domains. So PDK1 is going to phosphorylate and activate protein kinase B. So here again, we talked about JAK-STAT receptors earlier. It just goes a little more in detail, but nothing really new. So once your hormone binds to the receptor, it's going to activate your JAK-STAT receptor. So step 1, receptor binds to the cytokine, dimerizes, and binds JAK. So JAK is your Janus kinase, so they're going to phosphorylate each other in step 2, and as soon as that happens, it's going to phosphorylate the receptor also. So once the receptor is phosphorylated, that's going to allow the STAT to bind to it. So STAT is your signal transducer, activator of transcription. So once that binds to the phosphate, JAK is going to phosphorylate it again, and then STAT will dissociate and head to the nucleus where transcription can begin. So here we talk again about the serine-threonine kinase receptors. So we talked a little bit about this earlier. This just goes into further detail. So it's going to use TGF, which is transforming growth factor, and once that binds to your receptor, it's going to dimerize. So step 1, TGF binds to a type 2 receptor. Step 2, that type 2 receptor is going to phosphorylate the type 1 receptor. So the type 2 receptor is on the left, type 1 receptor is on the right. As soon as TGF binds to it, type 2 receptor will phosphorylate type 1 receptor. So once that happens, in step 3, you now have an activated type 1 receptor, which is going to phosphorylate RSMAD, and then in step 4, RSMAD is going to complex together with COSMAD, and then they're going to head to the nucleus where they're going to either activate or inhibit transcription. So here might just be good to know the names of the people who studied those G protein-coupled receptors. So remember earlier we talked about heptahelical receptors. Remember, they're also called G protein-coupled receptors. They are a 7-helix membrane spanning domain. They have a trimeric G protein, and that trimeric G protein is composed of alpha, beta, and gamma chains. So remember, this is just activating a chain of cellular events through either the adenylate cyclase pathway or the phospholipase C pathway. So this acts through a secondary messenger, which can be cyclic AMP, IP3, or DAG. So activates a G, binds to GTP, inactivated G binds to GDP. So this is how G protein-coupled receptors work. In step 1, a hormone has to bind to the receptor. So your receptor is the yellow, and then your trimeric G protein is your alpha, beta, and gamma subunits. So as soon as that hormone binds to your yellow receptor, it's going to cause a conformational change. This allows that G protein to bind to it. Also notice that there's a GDP in the alpha subunit. So in step 2, the G protein is going to exchange GTP for GDP, and that's going to cause dissociation of the G protein. So if you look to the right, the alpha subunit now has GTP on it because it exchanged it for GDP, and now it's by itself. So in step 3, there will be some sort of target protein along the plasma membrane. In this case, it's adenyl cyclase. And that alpha subunit with the GTP is going to bind to it. When that happens, it's going to make ATP convert into your secondary messenger, cyclic AMP. So in step 4, when GTP is hydrolyzed, it's going to become GDP again, and that's going to cause the alpha subunit to dissociate and go back to its original G protein that's bounded on the receptor. So that's what happens in step 5. So here's another important slide. It's a table, so make sure you know everything from this table. It's also your learning objective 7. So know the different subunits of heterotrimeric G proteins and what it stimulates, activates, or inhibits, as well as some of their physiologic uses. So on the left column, we have all our different subunits of that G protein. We have what it stimulates. So the first one, right, it's going to stimulate adenyl cyclase. And then we have their physiologic uses. So just be sure to read through all of these and be able to differentiate between all of them. So your heptahelical receptors, or GPCRs, can be grouped into 6 different classes based on homology and function. So class A, rhodopsin; class B, secretin; class C, class D; class E, which was cyclic AMP we talked about; and then class F. So just be sure to know what class represents what type of group. So here is the cyclic AMP pathway. So he focuses on this a lot. We've seen this already several times in the previous slides. So the first step, remember, is the hormone has to bind to the receptor. So it's not shown here, but once the hormone binds to a receptor, the receptor goes through a conformational change and allows that trimeric G protein to bind to it. So remember, your trimeric G protein is your alpha, beta, and gamma subunits. So also remember, your alpha subunit has the GDP on it. So it exchanges it for GTP, and that's when the dissociation happens. So now your alpha subunit with GTP is going to find a target protein. In this case, it's adenyl cyclase, so that's shown below. And as soon as it binds to adenyl cyclase, it's going to stimulate it to activate cyclic AMP. So cyclic AMP is going to activate protein kinase A, and then you're going to go through a series of phosphorylation cascades. So ultimately, you're going to get protein phosphorylation and gene expression. So here again is just showing you the pathway of what we just talked about. So if you look at the very top left, we have a hormone that binds to the receptor. That receptor goes through a conformational change and activates your G protein, so your alpha, beta, gamma subunits. Remember, your alpha subunit has GDP, so it can exchange it for GTP, and as soon as it does that, in the second step, it'll bind to adenyl cyclase, and ATP will switch over and become cyclic AMP, and then cyclic AMP is going to activate your protein kinase A. So that leads to phosphorylation of systolic and nuclear proteins. So same thing again, just illustrating that secondary messenger system. You can read through these. So here we talk about the phosphatidylinositide pathway. We talked about this a little earlier. Use this pathway for contracting and relaxing your muscles, and it deals with the PI3 kinase family, and it also involves G proteins and GTP. So if you look at the very top right, we're going to need inositol 4,5-bisphosphate. So that's also called PIP2. So remember from the previous slides, PIP2 can be cleaved by PLC, which is phospholipase C, into two secondary messengers. It can become IP3 or DAG. So if we follow that IP3 pathway, then ultimately we're going to lead to either relaxation of your smooth muscle or contraction of the smooth muscle. So he didn't test specifically or in depth on this pathway, but you should know that this pathway is going to be used for relaxation or contraction of your smooth muscles, and then what type of kinases it deals with. So we've been talking about plasma membrane receptors. Now we're switching over to intracellular signaling. So he doesn't talk too much about this, but he just wants to give you the chance to see the difference between them. So remember, intracellular signaling is dealing with your hydrophobic molecules. So they're going to be lipophilic hormones, steroid hormones, thyroxine, retinoic acid. So that's all you really need to know. So one of your learning objectives is to understand the termination of signals. So terminators are things that are going to completely stop the response. So some turn off quick, some turn off slow. There's many different steps. And if a signal is on for too long, then you could get certain diseases like cancer. So the picture to the right is what we've been looking at this whole chapter. So how does a chemical messenger work? First, you need a stimulus. That's going to cause a release of the messenger. Messenger binds to your receptor. Receptor sends a signal down, maybe sometimes through a secondary messenger, and then you're going to get a response. So everything that's in pink are your terminators. So he could ask, which of the following are signals of termination? So we have diffusion degradation, desensitization, downregulation, protein phosphatases, GTPases, phosphodiesterases. So all of those are examples of terminators. So here we have another clinical correlation. It's a learning objective to know the receptors and enzymes involved in both of the diseases. So the first one is myasthenia gravis. It's an autoimmune disease, which means your antibodies are going to attack your own body. So in this case, they specifically attack your acetylcholine receptors on the muscle. So remember, acetylcholine has to bind to acetylcholine receptors in order to trigger that contraction of your muscle. So if your antibodies are attacking your acetylcholine receptors, then there's less receptors for acetylcholine to bind to. So that's going to cause fatigue and inability to do repeated tasks. So numbers of acetylcholine receptors are greatly reduced because your antibodies are attacking it. And so for patients that have myasthenia gravis, we can give them certain drugs that inhibit acetylcholinesterase. And acetylcholinesterase is just something that comes out and eats up all the acetylcholine. So when we want to relax our muscle, acetylcholinesterase would come out, eat the acetylcholine, and then the response would stop and your muscles would be relaxed. So in this case, since we have fewer receptors, we want to prolong the acetylcholine being bound to your receptors. So we're going to take a drug to inhibit acetylcholinesterase, and there we can have the acetylcholine bound to the acetylcholine receptors for a longer period of time. So that's going to give us a brief increase in muscle strength. The second disease is anorexia nervosa. So that's just going to cause you to have a lack of appetite. The reason why you're going to feel that lack of appetite is because your endocrine hormones mobilize fuels from your fat. So epinephrine promotes fuel mobilization. There's going to be different receptors on different cells. Insulin promotes fuel storage. So here's your last learning objective, identifying the diseases and disorders related to cell signaling. So be sure to know all of these different disorders. Cholera, for example, the cholera toxin activates your adenylyl cyclase, so therefore enzyme remains continually activated. Pertussis, same thing. So pertussis toxin also activates adenylyl cyclase, and therefore the message cannot be stopped. So just be sure to read through the rest of them: Graves' disease, mutant RAS proteins, and then some drugs. So here's a slide showing all the different types of signal transduction pathways. He says you're not responsible for it, but if you want to look at the ones that you did learn, try to see if you can follow along and see if everything makes sense. So here again we have our disorder table. So myasthenia gravis, cholera, anorexia nervosa. Be sure to know the disorders and then also the details about it. That's it for Chapter 11.